And now it's with great pleasure that I introduce our next speaker, Dr. Csaba Cserep. He earned his PhD in theoretical medicine from Semmelweis University in Budapest. He now focuses in high-resolution 3D anatomical and functional studies at the Hungarian Institute of Experimental Medicine. And his projects deal with microglia, how they communicate, so the immunocomponent of the brain. And his current work explores how mitochondrial signaling contributes to the intracellular communication in the brain. So we are really looking forward to your talk, Csaba, and the stage is yours.
Thank you very much for the kind introduction. And I will just share my screen now. I don't know if you see it.
Yes, we do.
Perfect. So thank you for the kind introduction. And first of all, I would like to thank the organizers for this opportunity so that I can share our results and talk about these topics on this wonderful platform that you have been building. So as we heard from the introduction, I'm from Hungary and work in Budapest. And I joined the lab of Ádám Dénes in 2016. And basically, I'm coming from a neuroscience background where I learned a lot of high-resolution methods. And I was really shocked that in the neuroimmunology field, these methods were not really used at that time. So it was really an interesting building phase. So yeah, the last couple of years have been a bit hectic, but also interesting. So I will start with some simple introduction that we all know that the brain is a hyper complex system. It's really extremely complicated. And we absolutely need a holistic approach to try to understand this hyper complex system. So basically, the last decades, the understanding have been focused on neurons and the communication between neural cells, which is absolutely basic and core importance. But still, the brain is made up of lots of other cell types like glial cells, immune cells, and also a very beautifully elaborated vasculature. So this whole complex system needs to be approached with some holistic methods. So in this context, we try to understand the functional connectomics of microglia, which is basically the main immune cells of the brain. And I will have to make a short note here that until like the last decade, microglia has been viewed as a really passive component of the brain, basically just an immune cell sitting in the brain parenchyma, just waiting for something to happen, and without any how to say basic or normal function, and just waiting for something bad to happen, and then it could get in an activated state and phagocytose the dead cells and tissue debris. And in the last couple of years, lots of research have been done all around the world, which really show that the case is completely different. These cells really have very important functions in the resting state. And this is true even for brain development. So microglial cells have very important roles in brain development, but also in keeping the homeostasis and exerting neuroprotection. So this basic view of these cells has changed substantially in the last couple of years. So in this context, we really want to try to understand better the functional connectomics of microglia. And as I mentioned, for this, do you hear me?
Yes, we do.
Okay, sorry, it just froze. My computer froze for a second, but okay. So for this work to be done, we needed to gather a wide palette of microscopic methods, because a holistic approach doesn't only mean that we need to take all cell types into consideration, but it also means that we have to look at the brain on different scales, from centimeter scale down to the nanometer scale. Every scale possesses very important information that cannot be obtained at another scale. So we try to build really a methodological palette that spans all these scales. So first I will start with the introduction of this methodological palette, which is probably, it's not the I mean, not the very extreme um, microscopy methods, but I think these are put together in a really functional way so that we can get a lot of information out of our samples. And then after this technical introduction, I will go on the scientific part. So basically, we are using mouse and human fixed brain tissue. And the first step is that we use basic light microscopy. We use slide scanners just to evaluate the basic characteristics of different brain areas, and to evaluate the quality of our immunostainings. And then we go on to confocal laser scanning microscopy, which is a very important tool in our research. Probably it's not the newest development in microscopy, but still at this level we can get a whole amount of information out from the tissues. So here basically you can see the cyan-colored glutamatergic neurons in mouse neocortex, and in green we can see microglial cells which are touching the neurons at the dendrites and cell bodies as well. And normally we use a pixel width between 50 and 200 nanometers. And also we try to work in 3 dimensions. So we usually do large stacks, and we can label at the moment 4 channels in a parallel manner. But we will try to push this up at least to 6 parallel channels. Yeah, but there are some subcellular receptor distribution like information that cannot be got out with confocal laser scanner microscopy. So we turn to super resolution microscopy. And at the first time, we started to establish in our institute the STORM, this stochastic optical reconstruction microscopy, which means that which is basically a single molecule localization microscopy. The individual fluorophores can be localized, and then the image can be reconstructed. And we were first really happy that we could make a really nice image of a glutamatergic synapse here. The green immunolabeling is for vesicular glutamate transporter, which is the presynaptic side, and the red is the Homer, which is a postsynaptic protein. So at this 100 nanometer scale, we can see that it's really nice. So we were really happy about it. But then we went on that, okay, probably we would need to count these localization points and then try to get something meaningful out of it. And this is how basically a region of interest looked like. And what you can see very easily that the middle of the image is much more brighter than the sides. So the illumination is absolutely uneven, which is a big problem, because if I take one synapse from the middle and one from the side of the image, then basically these two synapses will look like this in the image, but in the biological reality, they can be very, very similar, which means that this imaging method, at least on that setup we used, wasn't really quantitative. So then we saw that some stormy clouds are coming to this method. So the uneven illumination that I mentioned, also the nonlinear signal, problems with multiplexing, channels mixing together. For this type of imaging, we needed a huge laser dose. Also, another big problem is that for this type of imaging, we needed a very aggressive mounting medium for the samples. And this very aggressive enzymatic environment in this medium basically destroyed our samples in a few days. So basically, we imaged the samples and then discarded them into the rubbish bin. So it's also very problematic. And again, the registration between the confocal laser scanning microscopy channels and the storm channels is not really working because of optical aberrations. So then we turn to the STAT super resolution microscopy, which is a completely different type of animal. It also very similar to the confocal laser scanning microscopy. It basically scans the whole sample. Uh, the big difference is that our active point spread function, the active effective PSF, is much more smaller than in confocal microscopy. So here is a microglial cell. With the green, we can see the calcium binding protein IBA1, which is specific for microglia. And in cyan, we can see the P2Y12 purinergic receptor, which is a microglia specific purinergic receptor. These two colors are in confocal. And also superposed, we can see the white STAT super resolution channel. So here are these two brackets which are enlarged to the right. So we can really see the difference. Either we are looking at a microglia process or the cell body and the process coming out of it. With the confocal channels, we can see that this immunosignal is really nice and it looks really homogeneous. But in reality, with the STAT super resolution microscopy, we can see that these cell surface receptors are forming nanoclusters, which can have functional importance. So these were the first images we made, and we were really happy that that as this scale on the scale which we can work with this STAT super resolution microscopy has really a meaningful addition to our measurements. So here is a sample from a human neocortex, again microglial process. We can see really nicely the cytoplasmatic calcium binding protein in green, and again the cell surface receptor in red in the confocal channel, and the very same fluorophores are imaged in the STAT channel, and we can really see that there is a stark difference between areas of these processes, and the very thin filopodia and the ending of the filopodia is really densely packed with these receptors. So now we are trying to do some functional experiments and compare how this receptor expression and clustering changes, because we really think that this will have a functional consequence on these cells. So here we can again do 4 channels in confocal, and we can do 2 parallel channels instead. And the pixel width is down to 16 and 20 nanometers. And yeah, the next step on our scale is obviously electron microscopy. But what we used for ages, like the conventional transmission electron microscopy, is a very nice method. But the problem is that we cannot really image larger volumes. So now we have, fortunately, a newer addition, scanning electron microscopy array tomography. which is basically done by my wife, and she cuts really, really long serial sections from resin-embedded tissue, and then we are taking them onto these silicon wafers, and these wafers are going into the scanning electron microscope, where we can make basically big volume areas and with kind of low resolution, and we can use these maps to really choose some regions of interest. And then we can put back these wafers in the scanning electron microscope and scan them with larger resolution. And here, for example, we can see, so the interesting part is basically the small silver intensified immunogold particles. So we can see here a microglial cell, which is wrapping around a capillary, and also with the processes, it is touching two neurons. And these basically the pixel width can be shrink down to 310 nanometers, but only we can label one target with this method. And the data volume is getting bigger, that's for sure. And then, as I mentioned, the next step is conventional, traditional transmission electron microscopy, with which we can really nicely see these receptor clusters again. These are the very same cell surface receptors on a microglial process. And we can see again really nicely these receptor clusters. And if we want to go even further, then we have transmission electron tomography, with which we really can look into the nanoscale architecture of these biological tissues. So here we can see basically a synapse with the anchored synaptic vesicles, mitochondrion with the internal crystal structure. Also, in a lot of cases, we can see the phospholipid bilayer itself. And on the postsynaptic side, we can see the postsynaptic density and also some tethering proteins. So for our questions, our scientific questions, this resolution is absolutely the maximum that we need. So we can go down to 0.3 nanometer isotropic voxel size with this method. Yeah, what about in vivo? So this was all fixed tissue, but we also need to do lots of functional studies. So we have a functional ultrasound setup. Also, we are working with miniscope imaging and also in vivo 2-photon imaging. So we think that if these functional methods can be coupled with the anatomical methods, then we have the chance to be able to answer to our questions, basically. So basically, this was the methodological palette we have been building in the last couple of years. And then I would switch to the scientific part, a bit more scientific part. So first, I would like to talk a little bit about microglia neuron somatic junctions, then microglia endothelial cell direct contacts, and just a few sentences about the role of mitochondria in intracellular communication. So the somatic junctions. So as I mentioned earlier, microglia have complex neuroprotective and homeostatic roles. But the underlying microglial interactions with neurons have been unidentified for a long time. And basically on this field of research, the main focus have been put on the interactions between microglial processes and neuronal synapses. However, it is very difficult to imagine that microglia would be able to efficiently monitor and dynamically control neuronal functions via these distant and heterogeneous and also dynamically changing contact types, because as we know, synapses through plasticity mechanisms can change really dynamically. So we hypothesized that there could be another interface, a more direct interface between microglia and the central part of the neurons. So basically, what we did here is that our colleagues used in utero electroporation so that we could label with red fluorescent protein a subset of cortical neurons in mice. And they did it in a specific mice line in which all microglia express green fluorescent protein. So this enabled us to use in vivo two-photon microscopy on anesthetized or even freely moving, not freely moving, but awake animals to look the interactions of microglia and neurons in real time. So what we see on these two-photon images that there are really microglia processes touching neuronal cell bodies. So then we made some measurements and compared the lifetime of these connections with the lifetime of other microglia connections established with the dendrites of neurons. And we found that the somatic contacts lasted 3-4 times longer than the dendritic ones. But what was even more interesting, that these microglia processes were touching the very same spots on the neuronal cell body again and again, as if there would be some connectors. So then we went on to try to decipher the molecular composition of these connections. And this is some early storm super resolution microscopy image where we didn't have the opportunity to use the STAD. So what we see here is basically the green microglia process is touching a neuron, which the membrane of the neuron is labeled with the red immunofluorescence. So what we see here is that the neuronal membrane is labeled in the storm channel with the yellow dots, and the blue dots are the P2Y12 receptors on the microglia process. So we can see that there is really, really small distance between these clusters, suggesting that probably this could be a direct membrane interaction between these two cells. So then we went on to use electron microscopy, and we found that indeed these interactions between microglia processes, which we can see in yellow, are really direct membrane interactions with the P2Y12 receptor, the black dots, really gathered to the connection points. And we also found that intracellular architecture of these connections from the side of the neurons is really interesting, because mitochondrial-associated membranes, even some mitochondrion-derived vesicles are anchored down to these connection points. And it was really logical to see this arrangement, because mitochondria are producing ATP, and the P2Y12 receptor is a purinergic receptor which can sense ATP and ADP. So this really made sense in a functional way. So without going into much details, we made a lot of functional experiments, and we found that microglial cells are in a perfect position in these connections to check neuronal well-being. And if there is a hyperactivity of the neurons, or any noxious stimuli happen to the neurons, then this contact, the coverage of this contact, so the surface, the touching surface between the two cells, is increased substantially. And on this increased surface, the microglia tries to perform some neuroprotective functions which we do not understand at the moment. But what we can see is that some bidirectional communication happens here. And if the neuroprotection was successful, then restoration can happen. But if the neuron is really heavily damaged, then this coverage can even grow further, and the microglia cell can phagocytose the dying or dead neuron. So here is a really nice example. Here, basically, one of my colleagues injected a neurotopic virus, and we can see here the blue color is basically the virus-infected neuron, which will be dead in like 6-10 hours. But at this early stage of the infection, of the viral infection, a green microglial cell is trying to wrap around this infected neuron. And in magenta, we can see the CD68 positive phagolysosomes gathering around. So really, this neuron will be eaten by this microglia, and we could do some correlative imaging. So here we can see the confocal images and the reconstructions from the confocal images. But after the confocal imaging, we process this sample even further, so we could find it in the electron microscopic image, the very same cell. And then we could even process this further to electron tomography, where we can see the connection, the direct connection between the two cells. Okay, so the next part is the brain development. So we thought that probably during brain development, microglia can also somehow influence neurons. So we gathered embryonic tissues, and also we checked on the right the adult neurogenesis. So we were looking whether these connections are present on developing neurons, and we found a very similar type of connections. And here again, with the correlated confocal and electron microscopic images, we can see the very same neuron that we labeled with double cortin. This is a post-mitotic neuron or neuronal progenitor. And also we found that microglia processes are touching these neurons. And also mitochondria seemed to play a role in these connections. Again, without going into details, we found that these connections are prevalent during embryonic development, and also during postnatal neurogenesis, adult neurogenesis. The morphological and biochemical buildup of these connections is very similar to the adult ones. And when we perform acute inhibition of these receptors, then the formation of these contacts is diminished. And if we genetically delete these receptors, then it will lead to disturbed cortical cytoarchitecture. So as this image shows, microglia really has abundant types of connections with other brain cells, like synapses, neuronal dendrites, the somatic junction I've been talking about, axon initial segments, Ranvier nodes, oligodendrocytes. But there is also a very interesting point that the neurovascular unit is also a very important part in the brain, which is built up of endothelial cells, pericytes, astrocytic end feet. And when we were looking at publications and schematic drawings of the neurovascular unit, the microglial cell was usually drawn there on these images, but without touching anyone. So we were looking, we tried to look how this cell can form connections with the vasculature. And yeah, just one sec. Just back to one note to the neuronal part, which I forgot to mention, that we really see that through these somatic junctions, these microglia cells are basically plugging their cables into the neurons and checking them if everything is all right. And if there is some problem, they will try to repair the neurons. And if it's not working, then basically they are there to collect the dirt. So yeah, now turning to the endothelial part. So how can microglia influence the vasculature, and what kind of connections can be built between these two cell types? So here in the first confocal images, we can see the microglia cells in green. The endothelial cells are labeled with the red lectin, and the astrocyte and feet are labeled with the aquaporin 4 channel, which can be seen in the light blue. And on the right side of the image, we can see different image planes from a confocal stack, and we can see that basically there is a microglia process which is um, basically touching the endothelial cell in between the astrocytic end feet. And it makes sense because the astrocytic coverage of these endothelial surfaces is not complete. So we saw some windows on these uh, astrocytic end feet coverage, and we saw that that microglia processes are touching directly the capillaries through these windows. And this was published in the Journal of Experimental Medicine 2 years ago. And we also went on to use electron tomography, and we also found that these direct interactions between the microglia process um, is again happens at places where mitochondria in the endothelial cells are enriched. And also the P2Y12 receptors, which can be seen here as the black silver particles, or in the reconstruction, the white spheres, are enriched on the contact side. So it was really interesting to see that a few days ago, paper came out uh, where the authors see a very similar effect, that microglia contact the cerebral vasculature through gaps between astrocytic and feet. But the problem is that, and I have to mention this, that they claim that basically their work is the first to see this, although we published it before. So I think it's a really difficult case. Anyway, so we saw this direct contacts, but also the endothelial cells are covered from the outside with the basal membrane. And we got the questions whether at these contact sites, the basal membrane are still there or not. And in these images, we could label the microglial cells with this cell surface receptor. And this labeling is so abundant on these contact sites that it's really difficult to see whether the basal membrane is there or not. So we thought that probably it's thinning, but I cannot really tell that whether it's there or not. So we made another staining where we used a cytoplasmic protein in microglia. And basically, this was the IBA1. And here in the green, we can see the microglia process. And with red, we can see the basal membrane, but we also can see that there are some places where there is absolutely no space between the cell membrane of the microglial cell body and the endothelial cell. So with this measurement, we could really show that there are parts of these connections where there is absolutely no basal membrane between the two cells, and it's really a direct membrane-membrane interaction. And then we also used the STAD, super resolution microscopy, to show that again, at these windows on the astrocytic end feed, basically the P2Y12 receptor positive microglia processes are really peaking in and touching the vasculature. So this was basically the endothelial part. And after all these contactomics studies, we really can just think of the microglia as the gardener of the brain, who is working every day to keep this beautiful garden working and functional. And then the last part of this presentation will be just a few sentences about the role of mitochondria in intercellular communication. So mitochondria are generally viewed as the powerhouses of eukaryotic cells. But they do much more than that. So in the last couple of years, lots of studies came out showing that these cells also function as signaling hubs, and they have very intricate signaling functions, not only in calcium signaling, but also in lot of other functions. And we thought that it must not be by chance that these very important signaling organelles are enriched in the connections we see. And this sample is from an embryonic brain, and this is a newborn. The blue one is a newborn neuron. And we can really see that the microglia is touching the neuron exactly where a huge mitochondrion is sitting in the neuron. So probably it makes sense because mitochondria are also involved in immune signaling, and they can like send out so many signals that can be meaningful for an immune cell. So probably this idea could make sense. So again, we are turning now to scanning electron microscopy, and with these reconstructions, we will be able to really see the positioning of these mitochondria within the neurons and also in microglia at these connections. So at these images, we can see that in the neurons, we see two colors of mitochondria, the yellow ones and the red ones. This is basically just labeled by us. The red ones are those mitochondria that are very close to microglial connections. So here, for example, again, we can see that we labeled those reconstructed mitochondria in red that are really close to the microglial connection points. And probably, maybe we will find something that these mitochondria are probably a bit different than the other ones. For example, we will be able to use STAD super resolution microscopy to study the suborganal distribution of mitochondrial proteins. So this is really just a starting point. So finally, I'd like to thank all the lab members and also my family for their help, and also the funding bodies. And I would like to thank you for your attention.